Hello everyone, we are from Group 2 Biology. My name is Zafira Pan Adeline 21213.
My name is Aijan Sadiko 20201.
My name is Hakun Dui 21202.
Now we are going to present our research titled Vaccine and Therapeutics Design through Structural Analysis of SARS-CoV-2 and its Variants. To start with the introduction, SARS-CoV-2 was firstly identified in Wuhan, China at the end of 2019. As previously studied, the virus is zoonotic, meaning it is transmitted between animals and humans. Here's the schematic diagram of the SARS-CoV-2 structure and its binding to human ACE2. The virus gains entry into human cells by using its spike protein to bind with human ACE2 as the cellular receptor. This is how it looks like when we design the inhibitor, so it will block their binding. Now I will explain the interaction between SARS-CoV-2 spike protein and human ACE2 receptor. In figure 2a, we have the spike glycoprotein trimer colored in green, blue, and yellow, and also human ACE2 colored in pink. Here is the zoom version of their binds. We labeled and highlight the key residues in respective color, and also the key residues are shown in sticks. After we study this, we will design an inhibitor to block the binding between them, thus preventing virus infection by using the folded program.
So as you can see on figure 3a, we've made a graph showing the progression and the immersion and the immersion of each strain of COVID, and then from alpha B117 to Centaurus BA275, and we've got all their picture, their graph here, and with all of their mutation highlighted. And as you can see, they progressively, as they the later they emerge, they accumulate more mutation. And the mutation mostly would appear in this yellow region that we call that is the RDB region, where the virus would use to bind to the ACE2 receptor, thus proving that those regions are vital to the entry of the cell, and those mutation would help help increase the infectivity of the virus or the cellular or the vaccine evasion. We've also, using PIMO, we've also managed to graph out 4 complexes with different therapeutics that can block spike protein in the research binding region. And for the first 2 graphs, we get the 2 spike protein antibody complex. First one is CTP59. Second one is STE90C11, and they both bind to the RBD region. And same goes for the next one. It's actually a nanobody complex, and the name is H11H4. The last one is not even a it's a peptide, and all of them just bind to the RBD, because then if the RBD is occupied, then it cannot bind to the ACE2, thus inhibiting the virus.
So now I'm going to talk about our future plans. As we already got to know, the receptor binding domain of SARS-CoV-2 binds to ACE2 receptor, which is important for viral infection. So the nature interaction of RBD and ACE2 not only is important for the understanding the initial step of virus infection, but also provides the necessary information to guide the effective development of drugs or vaccines. So the RBD is a key determinant for viral replication, and also is known to be an immunological main target for SARS vaccines with little or no antibody development enhancement. But the efficiency of conventional therapeutics, such as antibodies that block the viral infection, by targeting the RBD, is significantly reduced because RBD has a lot of mutations. So our future plans is to, we are going to design the peptide on folded standalone during our fall semester, and we are going to order the DNA and inject it to bacteria and let the bacteria express to protein. And after that, we are going to do the purification and PCR. And at the last, we are going to test the effectivity of the design peptide in blocking the bind between SARS-CoV-2 and ACE2.
That's it for our presentation. Thank you so much for listening and paying attention, and we will end it here.